Hello, in this video we're going to just review section 1.5 and 1.6. I've been wanting to get this done in class, but with lockdowns and camps and things like that, it's been a bit tricky. Now you do need to be aware, especially of 1.6, before we go on to our next unit, 2.7 I think it is, where we do transcription, translation and replication. That will be in the week that everyone's back from camp. So yeah, it's really important that you do watch this. For many of you, this will purely be revision. I have added a few extra bits in around some questions that I've been asked. So yeah, let's get going. Okay, in unit 1.5, so you did this as homework over the holiday. We looked at the origin of cells. So unit 1.5 here. And we talked about the first theories around the first cell and where it came from. Okay, and we said that generations ago they believed in this spontaneous generation. Okay, this was the leading theory amongst scientists for centuries, where they believed that cells just spontaneously appeared. Okay. This was until Louis Pasteur did his experiments and disproved the whole theory of spontaneous generation. Okay, here we've got Louis Pasteur. He was a French biologist and he did some experiments that showed or proved spontaneous generation to be incorrect. Okay, so this was an example of his experiment. Okay, he boiled some broth. Okay, so to neutralize it, so it was sterile broth. He then placed that sterile broth into 3 different containers, as you can see here in these setups. The broth was then incubated, so warmed over a period of time, and a sample from each flask was then transferred to a plate containing a solid medium, okay, so like a Petri dish or something, and then was incubated over some time, so it could be like a Petri dish with some agar in it. The only flask sample that showed the presence of bacteria was the open one, so the first one over here. Okay, why do you think that is? Okay, bacteria could get in through the open nozzle, whereas here it's sealed shut, and here it's got a water reservoir, so bacteria can't get in. The other 2 didn't show any evidence of bacterial growth. This indicated to Pasteur that the concept of spontaneous generation was incorrect, because if spontaneous generation was correct, all of them would have had bacteria in them. Okay, so a practice question here based on Pasteur's work. So how did Pasteur's experiments in sterile broth in open flasks demonstrate the principle that cells only come from pre-existing cells? And that was by demonstrating that the sterile broth would not ferment in contact with air alone. As it needed more to be able to ferment. Okay, so steps involved in the production of the first cells. So there's 4 parts to this. The first one is the production of carbon compounds, such as our sugars and our amino acids. Now all this would probably mean a bit more to you now that we've done our start of our our unit 2, topic 2, sorry. Okay, so thinking about what we're looking at, the steps involved in the production of the first cells. So first of all, we needed to get carbon compounds such as sugars and our amino acids. So Stanley Miller and Harold Urey created an experiment in which they managed to pass steam through a mixture of methane, hydrogen and ammonia. This is designed to simulate that first atmosphere on our Earth many years ago. With some electrical discharges that would have simulated lightning, they managed to called amino acids and carbon compounds that are required for life. Okay, so they were formed through this experiment. So we had this toxic environment on Earth before there was life. Methane, hydrogen, ammonia all mixed in the atmosphere, and it was the atmosphere had a lot of electrical energy in it due to the friction, due to heat and that area. Okay, without going into depth to that, there was a lot of electricity in the atmosphere, and that is what allowed the creation and the fixation of those elements to form our amino acids and our carbon compounds. Alright, so we've got our carbon compounds and our amino acids. Now we need them to start forming our polymers, so our long chains. And it's believed that this started to occur deep in hydrothermal sea vents. Okay, now these vents are gushing hot water, okay, and carrying inorganic chemicals like iron sulfide. Now these chemicals are readily accessible source of energy for the assembly, because remember we need that energy to assemble our carbon compounds into polymers, to develop our phospholipids, etc. Alrighty, so we've got all these compounds and these polymers being formed, but next up we need to be able to enclose things, so the formation of the phospholipid membranes had to occur. Okay, and if the phospholipids, they're one of the carbon compounds, but they've got to form into these bilayers, and this would have to then allow for the chemistry in the cell to be different to that out of the cell. Then, okay, so we've got cells, we've got a different internal environment to that that's outside, but now we need to develop some mechanism for inheritance, because if these cells are going to divide and grow, okay, and grow in numbers, they've got to pass on that genetic information. So DNA had to be developed. Okay. Now enzymes are required for DNA to be able to replicate, so we needed to get enzymes needed to be produced. Enzymes, remember, are made out of proteins. So which came first, the chicken or the egg, the DNA or the enzymes? Well, RNA may have been the earlier mode of this inheritance, as it stores genetic information. It is self-replicating, but it can act as an enzyme itself. So RNA potentially came along before DNA. Remember, you are the future scientists. You could be the ones proving or disproving that. Okay, so we've got our cell. We've got cells, single cells developed. Okay, we've got the compounds required for them. We've made the phospholipid membranes, and we've worked out the genetic information, how that's going to pass on between cells. We're looking at single cells at the moment. So we're looking at where these first eukaryotic cells came from, and we're looking at the endosymbiotic theory. Okay, now you should know that symbiosis is a relationship between two organisms where they require each other to live. Okay, so to exist, they need each other, and there's different types. There's communalism, mutualism. Okay, but we're looking at a similar thing now with this endosymbiotic theory. Okay, now, but how's this related to cells? Let's see. Okay, the mitochondria were once free living prokaryotic organisms that had developed the process of aerobic respiration. So once upon a time, mitochondria existed without the rest of the cell around them. Larger prokaryotic cells couldn't undergo aerobic respiration. So what did they do? They engulfed the mitochondria by endocytosis. Instead of it killing and digesting the mitochondria, I thought, hang on, this works. This organelle produces energy. So, rather than digesting it, they allowed the mitochondria to keep on living and thus produce some energy. As long as a mitochondria could grow and divide as fast as its host cell, it could live inside this larger cell indefinitely. Nature's clever. Okay, so this relationship, as I said, is the symbiotic relationship, as both organisms are living together, and as long as they gain benefit from the arrangement, it's called mutualism. So it's a mutualistic benefit. The mitochondria has got everything it needs to produce its energy within the cytoplasm of the cell, and the cell is obtaining energy from the mitochondria. Win-win situation. This is also true for the chloroplast. It's another example of an endosymbiotic relationship. Okay, if we just look at the mitochondria and the chloroplast, they have their own genes on a circular DNA molecule like prokaryotes. They have 70S ribosomes. Remember, that's a typical feature of our prokaryotes. They can transcribe their DNA and use the mRNA to synthesize some of their own proteins. We'll look more in that in our next unit, so don't panic. And they can only be produced by the division of pre-existing mitochondria and chloroplasts. So one mitochondria only come from other mitochondria. Okay, so that exam practice then on our endosymbiosis and that whole unit 1.5. Which statement provides evidence for endosymbiosis? Alrighty, let us see. Okay, B. Eukaryotic mitochondria and chloroplasts have their own circular DNA. Okay, that's the one. That's what we've just mentioned. Okay. Look at the question, because you look at that, experiments by Miller and Urey, you might think, oh, hang on, we did that, it would be that one, but it's not actually answering the question that's been asked. Okay, here we've got some past exam questions. We've got the electron mitographs show a typical prokaryote and a mitochondria. Okay, compare and contrast the structure of a typical prokaryotic cell with that of a mitochondria. 4 marks. Compare and contrast. How are they similar? How are they different? Using different examples. So if we have a look, the differences, you can say prokaryotics has cell wall, but the mitochondria does not. Okay, mitochondria. You can use B. Now with B, you can say that or that. C, you can say that or that. And then similarities, you've got 3 differences you could say, and then you've got 4 similarities. But remembering that you're not going to get a point just for saying an opposite. Okay, what other questions do we have? Explain how the mitochondria could have been formed from 3 living prokaryotes. 2 marks, and you're talking about that endosymbiotic theory. Let's go and see what they want you to say for E. Okay, so you're talking about the mitochondria being engulfed by a larger cell. Okay, and then making sure you make one of these points as well in that. Okay, and interestingly for that question, there is an examiner's report. So question A, answers were spread over the whole of the 4-mark range, and the scale bars allowed the sizes to be calculated. They were close, but not even so. A substantial number of candidates claimed that one was significantly larger. Okay, and B, this is generally well answered, but candidates explaining how endocytosis would have got a free living prokaryote into a larger cell, and because it made itself useful, there was a selective advantage in developing a mutualistic relationship rather than digesting the engulfed cell. Some candidates gave valid evidence for endosymbiosis, like the presence of DNA, 70S ribosomes, or double membranes. This was all things that you should have done over the holiday for holiday homework. Okay, now we move on to cell division. And this was unit 1.6, again another unit that was holiday homework. Okay, and we just introduced reminding you about mitosis, okay? And mitosis is the way that cells divide, okay? The process of cell division and how two daughter cells are generated from a parent cell, okay? Now the two daughter cells have genetically identical nuclei, so identical genetic information. So basically, this is the process that enables the cell to clone itself, but for this to happen, the DNA must be replicated, and that's what we're going to look at in our unit next of 2.7. Okay, so each chromosome is converted from a single DNA molecule into two identical DNA molecules called chromatids. These chromatids then become separated into different daughter cells. Mitosis is broken up in 4 stages, which we're going to look at in a moment. Mitosis is only one component of what we call our cell cycle. Okay, so here we have our cell cycle, and the key events that occur during the cell cycle can be divided into 3 main categories. We've got what we call the interphase, mitosis, and cytokinesis. Let's look at them all a bit further. Okay, during the process of mitosis, we've got our interphase, our prophase, metaphase, anaphase, telophase, cytokinesis. Okay, you might be able to make a little anagram to remember each process. Okay, so it's really important to remember that the interphase and cytokinesis are processes of their own. Okay, so if we're actually talking about mitosis properly, we're only referring to the prophase, metaphase, anaphase, and telophase. But the whole list of them, interphase, prophase, metaphase, anaphase, telophase, cytokinesis, is part of our cell cycle. You can make your own mnemonic to remember them. So here we have a diagram of mitosis, okay, and so we've got our cell cycle starting here. We go into our interphase, which is that first stage, not part of mitosis. Then we get into the prophase, metaphase, anaphase, telophase. So this is a cell, okay, and we're seeing what's happening within that cell. We're watching, okay, our DNA replicating, making chromatids. We're watching these spindle fibres form. We're watching the chromatids line up, the spindle fibres attaching to the chromatids and pulling the chromatids apart in the anaphase. Telophase, when 2 cells are then forming. One cell's carrying on through the cell cycle there. This cell is fully forming through cytokinesis. Okay, and on we go. Let's look at this in a little more depth. There we've got that cell cycle again. Okay, the interphase. Okay, so before mitosis occurs, this is a very active phase where processes are occurring. Our DNA is replicating in the nucleus. Okay, remember we're about to look at DNA replication in our next unit. Proteins are being made in the cytoplasm. Mitochondria and chloroplast numbers are increasing, remembering that they replicate themselves. The interphase consists of 3 parts, our G1 phase, our S phase, where the DNA replicates, and our G2 phase. Okay, so now we start mitosis. DNA undergoes what we call supercoiling, okay, so this is where it forms structures known as chromosomes. DNA naturally sits in a messy string-like form, a bit like spaghetti. Never call it spaghetti. Okay, and DNA needs to be arranged neatly so that cell division can occur. So it kind of gets compact and it curls up around these histones. You see the little proteins here, histones? Okay, so the DNA curls around them, becomes what we call supercoiled, and turns into our chromosomes. Okay, and you can see here in the prophase, okay, you can actually see the chromosomes there that have appeared. These are what we call our MTOCs. Okay, our MTOCs, these are the microtubule organising centres. Now you get these within the cytoplasm, so cytoplasmic MTOCs. You can also get them in the development of cilia and flagella in cells, but our important ones in this process are our cytoplasmic MTOCs. Okay, and their job is to form these things called spindle fibres. Okay, so basically what happens is our chromosomes are lying across the middle of the cell, okay, or the equator of the cell, and these spindle fibres, which are tubulin proteins and RNA, so made out of protein and RNA, they come out of the MTOC and they grab hold of the chromosomes, the chromatids now, and pull them apart. Let's not get ahead of ourselves. Alright, the nucleolus is broken down. Microtubules are growing from the MTOC and form our spindle fibres, okay, which link to the poles of the cells. So we've got linking to the pole of the cells and coming out of the MTOCs there. At the end of the prophase, the nuclear membrane is completely broken down. So we've got no nucleus. See, this is where the nucleus would have been here. Okay, now, still in the prophase, we've got two diagrams here, the early prophase of the nucleus, the late prophase there where the nucleus is broken down, our spindle fibres have come out to the chromatids. Okay, so from the prophase we move into what we call our metaphase. Now the chromosomes have lined up along the equator of the cell. So you see them all there, they're fully lined up. So in the prophase this was all just starting to occur. Now we've got this alignment. The microtubules now continue to grow and finally attach here to the chromatids, or to half a chromosome here called a chromatid. Okay, the little legs are called the chromatids. Alright. So they grow, the microtubules come out, they're going to attach to the centromere on the centre, and they're going to eventually pull this chromosome apart. Alrighty, the spindle fibres are also holding the MTOC to the cell wall there. Because obviously if they start pulling, you don't want the MTOC skimming into the middle of the cell. They've got to stay there and drag these chromosomes apart. There are two attachment sites on the centromere, enabling the microtubule to attach from each pole. Tension is then applied from the microtubules to ensure that the attachment's correct, because they've got to be fully attached to separate those chromosomes. Okay, so now we're in the anosphere. The anosphere. Don't know where that is, but we're in the anaphase. Okay, the MTOCs now contract the spindle fibres, microtubules, okay, so the fibres that they've sent out, centromeres on the chromosomes now divide because that force has been applied, and the two sister chromatids separate. Those spindle fibres, those microtubules, now rapidly pull those chromatids to the pole of each cell, so to each end of the cell, and this ensures that the same DNA, same copies of DNA is going to be in both cells that are produced from mitosis. Okay, so they've been pulled apart, and now in the telophase we're starting to reform the cell. The chromatids are now chromosomes. Chromosomes are pulled into a tight group near to the MTOC here. So you see how they're in that group near the MTOC, and a centre membrane is now forming. So a central membrane is now forming around them. The chromosomes will now start to uncoil, so they're going to go back into their spaghetti kind of formation. The cell is now ready to enter the interphase, where it's going to it could now redivide, okay, or cytokinosis can occur to make separate cells. Okay, so this occurs after mitosis, and the final separation now has 2 cells. It is different in plant and animal cells. In animal cells, the plasma membrane is pulled inwards, forming what we call our cleavage furrow, and a ring of contractile proteins, actin and myosin, inside the cell achieve the furrow. And when the furrow reaches the middle, it's pinched apart into the 2 daughter cells. In plant cells, vesicles all moved, so vesicles being, you remember in that first unit we looked at, they're just packages of fluid. They move to the equator, so we've got all these packages of fluid moving to the equator. They line up, pectin and other substances are brought in via vesicles and deposited between the 2 new membranes, forming our middle lamella, which will eventually form our 2 cell walls. The vesicles then bring in cellulose to the equator, deposited adjacent to the lamella, and we've got our 2 daughter cells. How can we tell if a cell is undergoing mitosis? And we're going to look at this under the microscope soon. Okay, we've got a ratio between the number of cells in mitosis in tissue and the total number of observed cells. We can see our mitotic index up there and how we would work that out. And we work it out using the equation of our mitotic index is the number of cells in mitosis divided by the total number of cells. Okay, just a quick look at cyclins. Now you understand what proteins are. These are proteins involved in regulating the cell cycle. They ensure tasks was performed at the correct time and that the cell only moves to the next stage of mitosis or when it is appropriate to do so. They were discovered by Tim Hunt accidentally. He was researching the control of protein synthesis in sea urchins and noticed an unusual pattern of protein activity. So further research led him to realise that the proteins were actually regulating the cell cycle, and he named them cyclins. Upon accepting his Nobel Peace Prize, Hunt spoke extensively about serendipity, a happy and unexpected discovery made by accident. Many scientific discoveries have been made this way. Okay, so in the video that you watched over the holiday, we then looked at tumour formation and cancers. Okay, and we said that a tumour was an abnormal growth of cells that developed at any stage of cell life in the body. If these cells adhere to each other and do not invade other tissues or move to other parts of the body, they're benign. But if these cells are abnormal and become attached and spread elsewhere in the body, they're classified as malignant. The development of secondary malignant growth away from the site of the original growth is known as metastasis. Okay, diseases due to malignant tumours are commonly known as cancers. Okay, cancer has many diverse causes. There's chemicals and agents known as carcinogens. The things that are mutagens are carcinogenic, such as some viruses, X-rays, UV rays, many chemicals. Mutagens cause a gene mutation, and mutations can then lead to cancers. Okay, so certain chemicals can lead to an increase in the risk of metastasis. Mutations are random changes in the sequence of genes. Okay, not all gene mutations lead to cancers. The genes that lead to cancers, if mutated, are called oncogenes. Okay, oncogenes regulate the cell cycle, and if mutated, the cell cycle gets out of control and tumours are then formed. We know that there are links between smoking and cancer. Okay, there's that positive correlation there. Alright, so past exam question. Let's have a look at this. In the micrograph, which letter points to a cell in the anaphase? Looking closely, you see, we've got cells there, and you can see here in the anaphase we've got those spindle fibres forming. Okay, that's what's giving that away. You can see those finger-like structures there. Yes. What describes nuclear division in stem cells? All we're doing is talking about cell division, so that's why the answer is mitosis. This was also homework over your holiday period, and that is all of that revision for 1.5 and 1.6. Thank you.